Today I wanted to introduce Matt Soldano. Matt was an undergraduate here at UCLA, and he started working in my lab as an undergraduate research volunteer, and then he stayed on as a staff research associate, and has been working with us over the past roughly 2 years. And the majority of his time is focused on the interpretation of DNA methylation data and trying to relate it to measures of health and aging. And so today he's going to be telling you about some of the fruits of that research.
Yeah, thank you, Matteo. Today is going to be a little bit more on inflammation. My name is Matt Soldano, and today I'll be talking to you about a non-invasive epigenetic measure of inflammation. So I'm going to try to unpack each of those words for you, starting with what do I mean by epigenetics? And so the epigenetics results I'm going to be focusing today, Matteo mentioned, were DNA methylation changes. And so, there's a few other different types of epigenetic regulation. There's histone tail modifications, there's RNA silencing, but DNA methylation, we feel like we can use as a very good marker for different measures of phenotype. And so for those who aren't familiar with DNA methylation, so we have these red pins here signifying a methylation site just upstream of a gene. And then we consider this to be a red light. There isn't going to be much transcription of that gene or the genes in that region because we have a closed chromatin state known as heterochromatin. So the protein that DNA wraps around is the chromatin, and when it gets packed tightly together, the gene transcription proteins are not going to be able, or the DNA transcription proteins are not going to be able to get into the genes in that region. And so that's why we consider it closed, and the genes in that region are not going to be able to be expressed. However, if the site was never methylated, or there's even pathways to unmethylate regions, then we have this open chromatin state, or what we call euchromatin, which is a green light. The DNA transcription proteins are able to get in there and transcribe the DNA and the genes that are in that region, expressing the proteins that those genes transcribe. And so we can view these methylation sites as a barcode, because barcodes, they're zeros or ones, and similar to that, the DNA methylation is either it's methylated or it's not methylated. And so we feel like we could scan the genome as a barcode and try to associate those zeros and ones with different phenotypes and with whether it's aging or diseases. And we feel like it's a pretty fruitful area of research. Now, what do I mean by a non-invasive epigenetic measure? So we utilize buccal swabs for the majority of our research lately. And so what a buccal swab is essentially a Q-tip, and then you rub it on the inside of your cheek, and you get the epithelial cells, so the cheek cells, as well as the saliva, so you can get a good mixture of granulocytes and epithelial cells. And also you could do microbiome analysis on it. So it's a pretty fruitful area for performing research on, because it has a lot of different cell type populations. And so we feel like we can get a good idea of what's going on, not just in the mouth, but also in the body generally. And so now what do I mean by inflammation? And today I'm going to be specifically focusing on the complement system for inflammation. And so what that is, it's a complex network of proteins that play an especially crucial role for the immune defense within our mouths. Since our mouths are a mucosal membrane where pathogens are frequently testing our immune systems, like when you're eating or even just when you're breathing, you're going to breathe in pathogens. Or if you're, riding down on your bike or going for a walk and somehow a fly ends up in your mouth, there's going to be constant pathogens that are going to be berating the system going on within your mouth. So another reason why we think buccal swabs are especially interesting. So this complement system is going to be a pretty frequent player, and it contributes to the overall oral health within 3 different functions. One of the methods is through opsonization, which is when the complement proteins bind to the surface of bacteria and other microorganisms, thus enhancing the recognition and destruction of the pathogens by immune cells just by tagging the pathogens themselves. Another way is through complement activation in the mouth, leading to an increase in inflammatory mediators, which are different processes that recruit immune cells to the site of infection, contributing to the initiation and regulation of the immune response. And then after the lysis of the pathogenic cells, another function of the complement system is the clearance and removal of debris and dead cells, as well as immune complexes, making sure that the immune complexes aren't attacking the host cells or just being in a chronic inflammatory state, which can cause damage over time. And so I've circled here C3, C2, and B. So these stand for the complement B3 protein, complement 2 protein, and the complement factor B. We can see that they're in this complex here where we have an antigen from a pathogen that's getting attached to an antibody from the host immune cells. And then we have this whole complement pathway with the C3, C2, complement factor B, as well as many other different proteins that lead to this cascade that cause the lysis. And so those are the ones that I'm going to be focusing on in my analysis. And that's why I circled them for you here today. So now, where do we acquire the data for doing our analysis? We've partnered with a fitness study that is trying to find what can we associate if we categorize people as fit or not fit. And by categorizing them, I mean by using their BMI as well as heart or blood pressure and a few other markers that have been known to be associated with people who are fit versus not fit, and seeing if we can really derive anything at a holistic level for the people that are fit versus not fit. And to do that, we have 47 people who are labeled fit, 46 labeled not fit, and we acquired their different measures of their fitness and health through BMI, measuring their METs minutes per week, which is just a marker for how much they exercise. We also got a comprehensive blood panel, which includes albumin, blood-urea nitrogen, among many others. We got some markers of what their habits are, whether they smoke a lot of cigarettes. We got a mental health questionnaire to try to see if there's a difference in mental health between fitness and being not fit. And we also acquired the metabolome and proteome of these people. For those who aren't familiar, the metabolome is just a bunch of carbohydrates that we can find circulating in our body. And then the proteome is the proteins that are going around in our body. And we're trying to use these as our metadata to try to predict them through targeted bisulfite sequencing of both blood and buccal samples. But today we're focusing on the non-invasive side. All of my results today are going to be on buccal samples. And this is very much fitting because it's the research in progress series. And so in progress, we are focusing on both the buccal and the blood, and going to compare the two and eventually get to maybe even combining them and seeing if there's a real difference between what we can predict between the buccal swabs and the blood. And another important component of what we were doing with the targeted bisulfite sequencing is cell deconvolution. And this is just a fancy way of saying that we're able to use a computer to determine how many granulocytes versus epithelial cells are in the sample. And this way, we can not only use it as a quality control measure. So if someone, for the buccal swabs especially, just put the swab in their mouth and it soaked up the saliva and we didn't get really any of the epithelial cells, we could be able to tell if that's going to be a problem. But we could also use it as a marker for metadata, because as you can imagine, someone who has an ongoing infection or a bad oral health, we could see that there's a lot more granulocytes simply due to the fact that that's what's going on in their mouths. And so we could use it also to predict the health of their oral microbiome. And from all of this data, so we're using the targeted bisulfite sequencing and cell deconvolution to predict this metadata using large data analysis and model creation. And to get to that stage, we have a 3-day full protocol about bisulfite sequencing. I'm not going to get too in-depth on this because it's not the focus, but it is a pretty long process of doing DNA extraction, library preparation, pooling and concentration. And then we get to the hybridization capture, which is an important step that separates us from epigenome-wide studies and targeted studies. And this is just to make sure that we have an increased resolution of the sites that we're really interested in. So instead of looking at seeing how methylated a site is once or twice, we can really sequence it in depth to get like 100 or even 400 measures of that same site to make sure that the associations that we're finding are actually significant and not just random. And then we do the bisulfite treatment, which is a very interesting method of determining whether the DNA is methylated or not methylated. I can get more into in depth in that during the questions. And then we do amplification and quality control. Then we finally get to the sequencing and the fun part, the data analysis. And so that's what I mainly focus on. But getting back to the hybridization capture and why it's a very unique and important method that we use. So CPGs, it's short for cytosine phosphodiester bond guanine. And this is because that is where the vast majority of DNA methylation is. And so we are looking at targeted CPGs. And we've acquired these CPGs from multiple different methods. The first one was from epigenetic clocks. I don't know if everyone's familiar with them, but it's becoming a very big growing field in terms of trying to predict not just chronological age, but biological age using these CPGs. They originally started with the Horvath clock, and then we had the Hannon clock, the Levine clock, the Omic-M clock. It's just constantly developing, trying to move away from just using DNA methylation to predict chronological age, but also to try to predict a biological age. So why do some people who are in their 70s look like they're in their 50s, and why do some people who are in their 50s look like they're in their 70s and feel like they're in the certain age group? And so it's trying to get to measure what is actually happening for the health of the person over aging. And so those studies have clearly had some CPGs that are associated with aging and biological age, as they call it. And so we feel like that could be very useful for our analysis. So we've used the literature to find those CPGs. And then we've also done some internal metabolism association studies, and those have to do with diabetes and other metabolism disorders. And we've been able to associate some CPGs with those different disabilities. And so we've used those in our analysis as well. And then lastly, the EWAS Atlas, the Epigenome-Wide Association Study Atlas, it is a repository where you can just search for terms such as obesity, age, inflammation, and other metabolism associations. And it will spit out a bunch of CPG sites that have been shown in literature to be associated with those different phenotypes or diseases. And so it's a very quick way to be able to find what's going on and some CPGs that would be very useful. And so using these three different methods, we've acquired 10 to 20,000 CPGs, depending on the analysis that we're doing, and are using those to predict the metadata that I mentioned earlier. So now the prediction component, the data analysis, where I live. So in the discovery process, I use MATLAB to perform linear regression and create models that we've measured using each of the methylation sites in our filtered probe set from the hybridization capture. And since it's filtered instead of an EWAS study, I like to call it a TEAS because it is a targeted epigenome association study. We're not looking at all the epigenome, we're looking at specific sites that I mentioned for specific reasons. And so this equation here, this is showing a very simplified version of what I'm doing in terms of age and sex correcting our linear regression models. And to simplify it even further, all we're trying to do is trying to see if methylation increases, does the amount of that trait increase depending on what that trait is. And so we have methylation here, and we're trying to predict it with certain coefficients for the trait time plus certain coefficients for the sex and certain coefficients for the age, because we really want to separate the age and the sex from the trait. We don't want to see, okay, yeah, we know that the DNA methylation is going to change between a 20-year-old male and 20-year-old female, or a 20-year-old male and a 60-year-old male. We know that there's going to be changes there. We really want to get down to what is the biological mechanism that is ubiquitous to everyone, regardless of their age and sex, for different traits and their methylation site. And so using this linear regression technique, we're able to determine the model p-values. So how significant are those models and associations? And then once we've found certain sites that are significantly associated, and when I say significant, it's not the normal 0.05 or less for the p-value. In these large bioinformatics studies, it's more common to use something like 10 to the -5, so 0.00001 or smaller, just because with these large data sets, it can get pretty easy to get small p-values, even though when you really dig deep into the analysis, they're not that significant. So we set a very high standard for our significance values. And then once we've done that, we can correlate and make scatter plots between the methylation and the trait to make sure that it's not due to any outliers and that there are actually very interesting associations between the site and the trait. And then once we have the sites and the traits that we're interested in, I then go to the UCSC genome browser. And this way I can just search those sites where they are in the genome and see what genes are located in those regions. And then go back to the EWAS Atlas and put in those genes and those CPG sites and see what associations there are between that gene and different phenotypes, as well as what associations have we seen with that CPG site and the phenotypes. Maybe there's some overlap, and so that's part of the analysis. And then to see if there's anything that isn't already in the EWAS Atlas, we do our own literature reviews, and this is very much an ongoing process of trying to find what has been new that has found any type of relationship between the gene that we found and any type of phenotype. And now, so the Manhattan plots, for those who aren't familiar, what they do is you put on the y-axis a -log10 p-value. So that makes it so that the larger the number on the y-axis, the smaller it actually is. So the more significant it is. And like I said, we usually use this cutoff for a 5, so 0.00001. And anything above this line, we consider to be a significant association. And here we have age and sex-corrected metabolic syndrome. And that is, metabolic syndrome is a combination of your waist circumference, your cholesterol, blood pressure, and a few other measures of is there something wrong with your metabolism? And we found one interesting site in chromosome 19 from our panel, and a few sites that were very significantly associated with this trait in chromosome 1. Again, ongoing, very much research in progress. We'll definitely get to analyzing those sites in the future. And then, as I mentioned with the epigenetic clocks, there are a lot of sites all across the genome associated with age, which is not surprising since our panel does use some of those CPG sites that it seems like we have enriched our panel for sites that are associated with age. But this is kind of why those epigenome clocks work, is because there's just DNA methylation all across the board that is associated with age. But now it's trying to understand, why are those sites associated with age? What's going on with those genes, and why are they being methylated over time or unmethylated over time, depending on the age of the person? So really getting into the nitty-gritty of understanding what's going on at a biological level, we started with that C3 complement protein that I mentioned at the beginning. So it's a part of the complement pathway. And we found that there are 4 sites here within about 1,000 base pairs, so very close together in the scheme of things, all in chromosome 19, that are very strongly associated with this complement 3 protein. And so, okay, we thought that was interesting. We'll do some analysis, but first let's look at a couple other traits. And then we look at this complement 2 protein. And again, there's some overlap here between these chromosome 19 sites and the associations between the complement 2 and complement 3 protein. So this makes sense. They're both under the same pathway. But now let's keep looking at what's going on in this region. We also have the complement factor B protein, another protein within that complement cascade associated with this same region within chromosome 19. We continue to look at this region, and we have fat percent, so the amount of adipose tissue a person has, and it's also strongly associated with this region of the genome. So we're trying to understand, okay, we have an inflammatory pathway and we have fat percent. There have been some association studies that have found that fat percent is associated with chronic low-grade inflammation. Now, what is going on here? Why is it associated at this specific gene, and why is it associated with complement factors? And so doing a little literature review, adipocytes, they are the cell type that we gain with more fat tissue. And what they have been shown to associate with, like I said, is chronic low-grade inflammation with secreting cytokines, chemokines, adipokines, and then it also does impact these complement factors. So maybe there's a link there. So we keep looking further and further, and we put all these sites within chromosome 19 next to each other, and we look across the board. There's a lot of correlations with these, specifically the complement proteins, and then we have fat percent, and we also have group. Like I mentioned at the beginning, we separated fit versus not fit people. As you'd expect, it does align a little bit with fat percent, but it is not one-to-one. But we can see that if we separate people into being fit versus not fit, there is a difference between their inflammatory pathways and methylation. And then I also mentioned at the beginning, we did cell deconvolution to look at the epithelial percent, and we can see that these sites are also very strongly associated with epithelial percent. So maybe there's something going on with the amount of granulocytes in the sample and how methylated those regions are and how that's associated with the complement protein. So there's a lot of different things that we want to link. What is this mechanism that's linking all of these specific methylation sites to inflammation? So we took all of these sites and we put them into the UC Santa Cruz genome browser. And we found that they cluster a little bit in this gene right here, and another also in this gene, but also a bit upstream of this gene. That the UCSC genome browser does list the promoter as being here, but what we found in literature is that promoters can extend for thousands of base pairs. And so this is still within just a couple hundred base pairs, very much in the realm of the promoter region. And so what is this gene? It is the peptidoglycan recognition protein. And what is that peptidoglycan recognition protein associated with? Well, it's associated with obesity. So we found, okay, there's an actual biological explanation for maybe why this gene is getting methylated and why is it associated with the complement protein. And so we keep diving deeper. Why are the complement system, immune cell percent, fat percent, all correlated with, but specifically an increased methylation of this PGLYRP1 gene? Why increased methylation? So to really quickly, before we get into that, we want to understand what this PGLYRP1 protein does. And it's a part of a peptidoglycan recognition protein family that, surprise, surprise, recognizes peptidoglycan, a major component of the bacterial cell walls. This recognition is critical for guiding neutrophils and macrophages towards potential infection. So maybe that's why some of the granulocyte percents are increased in the people that have this site being methylated more. And it gives us a very interesting idea of what's going on with this correlation. So taking this all together, this is our current working hypothesis. So under normal conditions in a healthy individual who's labeled as fit, there can be local inflammation due to either bad hygiene or just a stress response because of a pathogen. And so that's going to be associated with more PGLYRP1. There's going to be decreased methylation of that region. And so that way the local immune response can react properly and get rid of that pathogen. However, in people with increased fat percent, more adipose tissue, there's going to be a low-grade chronic inflammation due to those increase in adiposity and adipocytes. And that could lead to an increased methylation of those regions. So we think that the connection here between the inflammatory response from the complement pathway and the increased methylation is bridged by the adiposity. And we've even found some studies that have linked that PGLYRP1 specifically is downregulated with people with type 2 diabetes. So there must be some association and connection here with adiposity and the methylation of this region and general inflammation. Because on a similar vein as type 2 diabetes, where you have increased or chronic high glucose will reduce the insulin sensitivity for those people with type 2 diabetes, maybe something's going on here where with the chronic low-grade inflammation from the adiposity, you also see a decreased sensitization for the PGLYRP1 and having it be expressed in response to local inflammation. So we think we have a really good pathway, a mechanistic pathway that explains the methylation of this region to not only fat percent, but inflammation. So we could use this as a barcode for understanding not just the health of the person, but the inflammation and what's going on with their adiposity. And now I'd like to thank Matteo Pellegrini, not just for the introduction, but for really helping me along the way, develop myself as the scientist, and acquire all these data sets to really dissect what's going on with the epigenome and different phenotypes. As well as Mike Thompson, he's another staff research associate in the lab who's helped acquire all this data set, process it, and help me along the process as well. Thank you for listening. Does anyone have any questions?